So there we go. So I'm here to present the progress on the COVID Biorepository. So there are two specific aims for this project. Obviously, number one, to collect and store biological specimens, data from functional and anatomical imaging modalities, and clinical data and patient outcomes from hospitalized COVID-19 patients here at UCIMC. And the second specific aim is to make those collected specimens available to qualified investigators who will be performing research on predictive imaging and biomarker studies that can improve patient care. So we've really, as this project has evolved over the last couple of weeks, we have developed a workflow to maximize the flexibility of capturing specimens. And so, as I'll describe in item number 3, there is an IRB-approved protocol for prospectively consenting COVID positive patients, and we'll talk more about that. But in addition to that, there are also patients who generate clinical specimens that do not need to be prospectively consented. They generate discarded specimens. And so using a different blanket IRB that the experimental tissue resource runs under, we can collect those discarded specimens. And so investigators that don't particularly need access to protected health information, those types of studies can be serviced with these discarded specimens. And if an investigator does need particular subtypes of clinical data, that kind of data can be linked back to the biospecimens retrospectively using the ETR honest broker mechanism. And so the typical types of discarded specimens that have been collected thus far include plasma and anticoagulated whole blood samples from clinical chemistry, serum samples from the serology division within chemistry, nasopharyngeal swabs from microbiology, and we have an enormous number of negative nasopharyngeal swabs and a growing cohort of positive specimens, which to be for the most part, the positive nasopharyngeal swab specimens have been retained for clinical validation purposes, but as the number of platforms available for testing have increased, those specimens will be released for research purposes as soon as possible. And then from a human tissue standpoint, we've only had 4 in-house COVID deaths, and we did manage to consent the first of those patients for autopsy early last week, and we collected duplicate samples from 19 different organs, flash frozen and formalin fixed. So the discarded specimens are one pool of specimens. The other, or the second pool, are discarded specimens from other clinical trials that obtained their own IRB approval. So for instance, the remdesivir study enrolled 22 patients here at UCIMC, and clinical specimens in excess to those needed to be sent to the central lab for that will be made available for subsequent research pending modification of IRB approvals to use those samples. And then really the backbone of the effort here is enabled under IRB 2020-5783, in which COVID positive patients in-house will be enrolled. You can see the inclusion criteria and exclusion criteria there. And there will be a concurrent clinical database of clinical factors that will be collected concurrent with those patients' admission. And we'll talk about that data set in a second. So the COVID clinical database captures information about all of those types of specimen collections. The biospecimen records are collectively curated within FreezerWorks, which is a biorepository platform. We have a shared license between ETR and ICTS. For the prospectively consented patients, those patients will be registered in Encore, and an ECRF, an electronic case report form, will be generated in REDCap, and you can see the table on the right there that captures all of the different data fields that will be obtained about those patients. And I won't bore you with the details, you can see the list there. So just as a progress report, this table summarizes the totality of the discarded specimens collected as of Saturday. Just as a quick aside, Sahir kept using my name in the first presentation. What he meant to say was Delia Tifreya. She's the ETR manager who's done the bulk of the work here. She deserves all of the credit. I deserve none of it. I'm just here to report this to you. But what you can see is that we started collection on April 3rd. We've captured biospecimens from 83 separate COVID positive patients. Most of those folks were in-house and have been discharged. We have, you can see under length, we've got 10, 15, and 12 patients for whom we have collected less than 10 days, 10 to 20, and 20 days, or greater than 20 days of samples daily while those patients were in-house. So we have a number of sick COVID positive patients for whom we have serial plasma and whole blood collections. And so those will be very interesting patients for the subsets of investigators who need to track, particular biomarkers over the course of a hospital infection. And you can see under types, the specimens really shake out about 50-50 between anticoagulated whole blood and plasma specimens. So in all, we've collected nearly 1,000 specimens. And beginning on April 27th, as the pathology department rolled out a number of in-house serologic tests, we've now collected 223 serology specimens from leftovers in the serology division. And so Delia has been collecting on the order of almost 30 specimens a day. Those specimens do not come with clinical results, but again, because of the honest broker function, we've retrospectively gone back into EPIC and identified, we've reviewed 44 of those serology specimens, and 4 of them are seropositive. It's not a statistically significant sampling yet, but it gives you an idea of what we can do. So in all, we have available for investigators nearly 4,000 total aliquots of COVID positive serum and plasma and serology samples. So in terms of the, just quick last slide, the workflow for distribution of specimens. So we've centralized the specimen request process through the CRAFT COVID committee. So investigators apply to CRAFT COVID via that fund opportunity link. The proposals get reviewed. If the proposal gets approved, the specimen then secondarily completes a biospecimen request form, which goes to the newly formed biorepository subcommittee, which will review that biospecimen ask and match it with our inventory to make sure that that ask can be fulfilled. And then those specimens will be distributed to the investigator. So that's where we are, and I'd be happy to take any questions.
I thought this project was approved, right, Suzanne?
I'm sorry?
I thought this wasn't the biospecimen project approved under the CRAFT COVID funding?
Yes, yes.
So I don't know, on the last slide, it was said if approved.
Oh, no. I apologize. That workflow is for investigators who have, that's for all the other investigators who are interested in obtaining COVID positive biospecimen.
Oh.
And
Okay. I think I mixed it up. Okay, because you had the
Sure.
fund op and competition, and I thought, anyway, so,
I apologize. I didn't make that clear.
okay.
I have a quick question. Does this mean that the only way we can have access to the biorepository is essentially we have to apply for a grant through the Craft COVID committee and have that be reviewed and everything? Or like, let's say somebody wanted some small number of specimens for a small validation study, but it wasn't going to be highly costly. Like,
So I should probably let Daniela speak to that. But my understanding is if an investigator is interested in having COVID positive biospecimens, they don't need to.
And I'm happy to take the question.
Go ahead.
If you'd like to. So just to answer the question, no. The biorepository is open for everybody that is interested in specimens. In the beginning, there will be concerns that we will collect specimens and they're gonna not be enough for the projects that are already funded through our pilot. So what we said is that the first priority is going to be given to the projects that we have funded through the Craft COVID Craft COVID funds, because we didn't want to have projects funded to do the work and not being able to do the work. The second very close priority was any project that is funded through any kind of peer review. And then the third category was any project that our investigators are conducting or that, our investigators are conducting in collaboration with outside organizations, either academic or private. What we didn't want to have, if possible, and that's something to discuss later, is just to sell, quote unquote, specimens, or to give specimens outside of our institution without having meaningful collaborations.
Suzanne, Mike Bookmeyer. Rob or Daniela, do you have any plans to adapt as you listen to what some of the complications are of COVID, especially in children? This Kawasaki-like syndrome is very interesting. And it would be really, really critical to get tissue in a postmortem, certainly, from a patient who had shown that kind of symptoms.
Well,
So
I'd actually like to ask Rob, is there any different kind of a consent that you get pre-post-mortem, or is that just a standard consent?
It's a standard consent, and it's analogous to the universal consent that anybody coming to UCI for surgical procedures, they understand that any tissue that is in excess to that needed for clinical diagnosis may be made available for research purposes.
Okay, thanks.
And
I think Mike is asking something different there.
Yes, so let me take my questions, if that's okay with you, Mike. We want to create a biorepository that is as comprehensive as our researchers need. So your question will have to be addressed in combination with CHOC, because as you know, the Kawasaki disease is something that we see in children. So those will not be necessary patients that we see at UCI, that those kind of patients will go to our colleagues at CHOC. The easiest way is to make a request on the biorepository website, which has been publicized extensively, and it can be found with links both on the ICTS and the CCR, as well as on the School of Medicine Office of Research website. So multiple places, make a request, let us know why you need it, and then we'll be happy to start brokering for you for those specimens.
Thank you.
Mike, do you know what the incidence of that Kawasaki-like syndrome is in kids? Is it too early really to tell?
Do you mean by patient numbers or by newspapers that it appears in numbers? It certainly is appearing in multiple sources right now. We could find that out very easily with an email to Lise Borofsky in New York. I know her, and she's quite a good scientist and physician.
And I would say that that will align perfectly with Dan Cooper's interest on with Braid on the pediatric effect of COVID-19. So I think this is a discussion that's worthwhile continuing.
Yeah, Mike and everybody. So I just was contacted by NCATS about this very topic. They're putting together a national consortium. They've already reached out to the Kawasaki people, some of the leaders of which are, as you know, at UC San Diego.
Right.
And they call this syndrome PIM, the Pediatric Inflammatory Multisystem Syndrome. So we're just at the beginning of trying to figure this one out. But it's a great point, Mike.
If you need anybody to break any ice down there, I know Jane Burns quite well. We've collaborated before.
Right, she's on that group. Thank you.
Right, right.